Julian Thayer is an emeritus professor at The Ohio State University, and he is currently at the School of Social Ecology as a distinguished professor of psychological science at UC Irvine. And he has like a million awards and is way too distinguished to go into the whole bio right now. But you can, of course, read about him online. Just briefly, his research focuses on psychophysiological aspects of self-regulation, particularly parasympathetic influences on physical and mental health. He is a pioneer. I mean, anyone who wants to know anything about heart rate variability, Julian's like the first person they think of. Thinking about heart rate variability, his work's been published in lots of different places. I should say he has come out with a number of really interesting and thought-provoking theories. The latest one that I spent a lot of time thinking about is his generalized unsafety theory, which I find fascinating. We were supposed to have a fun fact you can't find on Google to introduce our speakers with. And actually, if you look up Julian, you'll quickly find out he's a very, very accomplished jazz musician. I actually have one of his CDs, which I'm showing you here. I highly recommend listening. It's really terrific. And my fun fact would be that Julian, who is really, really accomplished, was very kind. He came to visit Boston, and I brought him to our house for dinner. And my son, who is a bass player, Julian is a bass player, was super excited to meet Julian. And Julian was very kind and listened to him play and said nice things. So that's my fun fact. So kindness, along with accomplished musicianship and a brilliant scientist, all in one. So with that, I'm going to hand it over to Julian.
Thank you, Laura, for that kind introduction. I'm going to share my screen now, if this is possible. Let's see. Whoops, that's not where I want to go. Let's see if that works. Okay. I'm so excited to be here. This is really a fantastic initiative. I want to elaborate a little bit on Alyssa's comments about well-being and how I'm trying to think about it. So emotional well-being is not just the absence of distress, and it's not just being up to the challenges of life, a sense of mastery, and it's not just resilience, that is being able to take a challenge and going back to a baseline level, but it's the growth state where facing and meeting challenges makes us stronger. So in this sense, it's kind of that which does not kill you makes you stronger kind of idea. So this is what I'm hoping to convey to you, and that this type of emotional well-being makes the pool of resources that one has bigger. It just doesn't bring us back to sort of level. It actually allows us to grow. And this is not only individual growth, but societal growth. And this sense of mastery and safety leads to compassion, which is the ability to help alleviate the suffering of others. And ultimately, I think that's what the goal of this network is all about. And I'm so happy to be involved in it, and with the great colleagues all over the world and all over the country. So with that in mind, the question is, what might the physiological substrates or concomitants of this growth state be? And as alluded to, we're going to focus on the autonomic nervous system, but I want to state that the way that we characterize physiology into these sort of separate, you could call it silos or compartments, the body doesn't know any of this. This is only for our pedagogical purposes. The body just mounts a response. So all of these webinars are actually interconnected. So I want to emphasize the fact that even though we're focusing on the autonomic nervous system, all of these systems are interconnected. And that's why they're so important that we highlight each of them and then show their interconnections. So from the autonomic nervous system perspective, what might be the physiological concomitants of this growth state. And I'm going to focus in particular on the vagus nerve. And this is the reason why. So this is a quote from Charles Darwin's book, Expression of Emotion in Man and Animals, published in 1872. And what's interesting about this quote is that in this approximately 600-page book, this appears on page 72. So this is the basis of his work, not. So as his basis, what he states is, and he's talking about the work of the French physiologist Claude Bernard. Claude Bernard also repeatedly insists, and this deserves a special notice, that when the heart is affected, it reacts on the brain. And the state of the brain again reacts to the pneumogastric nerve, which we now know as the vagus nerve, on the heart. So that under any excitement, there'll be much mutual action and reaction between these, the two most important organs of the body. So that's what I'm going to elaborate on, this quote from Darwin, which was the basis of his book on expression of emotion in man and animals. So just a very brief introduction to the autonomic nervous system. It's thought that there are, the way that it's been characterized is there's a sympathetic division associated with fight or flight. There's a parasympathetic division associated with rest and digest. I want to emphasize that it's not just fight or flight. There's also a third F, which is freeze. So fight, flight, or freeze, which in a certain sense could be characterized by the parasympathetic division. But the larger issue here is that most organs of the body are dually innervated, and these divisions, again, are not known by the body itself. These are for our purposes. So now, why do I emphasize the vagus? Well, the vagus is the longest nerve in the body. Approximately 80% of the fibers are sensory. That is, there are receptors on the vagus nerve for cytokines, for glucose, for cholesterol levels. All of these things are then fed to the brain, and in particular to the nucleus tractus solitarius, and then up to higher brain regions. And then on the efferent side, again, these brain regions feed into nucleus tractus solitarius, which then feed into the nucleus ambiguus and the dorsal motor nucleus of the vagus to innervate these organs of the body and regulate these physiological processes. So the vagus nervous system is a psychophysiological system essential for the balance of the organism. And the vagus is really the controller in this system. So there's a left vagus and a right vagus. I won't get into all the details of the physiology, but just to give you a sense of where this vagus is going. And this leads to a concept of autonomic balance, which is a predictor of mortality and morbidity. It underlies a broad range of responses linked to what is called, our dear friend, departed friend, the great Bruce McEwen, called allostatic load. It's associated with the central nervous system, which is what I'm going to talk about today. And this may help to explain how psychosocial factors are instantiated in physiology and disease. And this is our key link to the emotional well-being. And it may help to explain known health disparities, though I won't talk about those in any detail. So in the context of this notion of autonomic balance, this is a figure from Steve Julius. When sympathetic tone is high and parasympathetic tone is low, there are a range of metabolic, trophic, hemodynamic, and thrombotic factors that are in play. And so let's looking at relative sympathetic overactivity can lead to insulin resistance, which can ultimately lead to diabetes and endothelial dysfunction and weight gain. Trophic factors are associated with the renin-angiotensin system, hyperinsulinemia, increased blood pressure, again, potentially ending in endothelial dysfunction. Hemodynamic factors associated with elevated heart rate, arrhythmias and tissue ischemia, thrombotic factors associated with platelet activation, thrombosis. So these are the ill effects. Now, the question is, if these things are so bad for us, why do we still have them? And the reality is that in the short term, these are adaptive. So in an unsafe environment where one has to be prepared to fight, flee, or freeze, uh, this relative increase in sympathetic tone is associated with in an environment where food is scarce, you want the calories you get to lead to weight gain. In a situation of fight or flight, you want to be able to mobilize the muscles to fight or flee, increase blood pressure, increase heart rate. And in the event of being injured in such an encounter, you want your blood to coagulate so you don't bleed to death. So in the short term, these are all adaptive responses, but in the long term, they can become maladaptive. You know, I often ask when I give this type of talk, if anyone knows what the average lifespan in the US was in 1900. And it's about 40 years. So a lot of these chronic diseases that plague us now were not a factor in death at at that point in time, and that the modern diseases are lifespan diseases, which are a result of this adaptive response being prolonged. So Laura alluded to some of the models that we have developed. One was the neurovisceral integration model, suggesting that there's a network of neural structures that can be indexed by a measure of autonomic nervous system function, the parasympathetic system in particular, which is heart rate variability. Higher heart rate variability is associated with greater prefrontal inhibitory tone, and this lack of inhibition leads to undifferentiated threat responses. So this is, again, a model that Laura alluded to, our generalized unsafety theory of stress model, which suggests that the default response to environmental challenges is a sympathetic fight or flight response. And it was adaptive. I'll say a bit more about that momentarily. So this is just a set of neural structures that are involved. This is the central autonomic network. This is from the neurologist Eduardo Benaroche. For our purposes, what's important is that the output of this network of neural structures goes via the stellate ganglia and the vagus nerve to the sinoatrial node of the heart. We know the pathway. Again, I'll skip a little bit of the physiology. We can come back to that later if you have questions. But at the sinoatrial node of the heart, there are differential neurokinetics. So the sympathetic pathway, which you typically associate with fight or flight, is actually relatively slow, order of magnitude of seconds. And if we relied on the sympathetic pathway to get us out of danger, we would all be dead. It's just too slow. The cholinergic pathway, however, is on the order of magnitude of milliseconds, the parasympathetic or vagal pathway. And the combination of these two influences on the heart lead to the complex variability that characterizes a healthy heart, and we call heart rate variability. Again, there's a left vagus and a right vagus. The right vagus goes selectively to the sinoatrial node of the heart. The left vagus goes selectively to the atrioventricular node. Again, this has some important implications, but I won't get into the details. You can ask me questions about this later. So the parasympathetic nervous system influences dominate. Our sinoatrial node cells beat about 105 to 115 beats per minute. And most of us, however, have a normal resting heart rate between 60 and 80 beats per minute. This suggests that the vagus is working and that the inhibitory influence over cardiac chronotropy, heart rate, by the vagus is substantial in most individuals. When that, however, becomes compromised, that leads to disease states, and that's what I'll further talk about. This figure from Warner and Cox just shows the dynamics of the vagal and sympathetic system. The vagal system is very rapid. So you stimulate the vagus, heart rate decreases quickly. You stop the stimulation, it increases rapidly. The sympathetic system, on the other hand, has a slow rise time and, importantly, a slow decay time, such that prolonged sympathetic activity beyond time of stimulation can be deleterious. And this is what we think this prolonged physiological activity leading to ill health is about. So in this context, heart rate variability refers to the time between heartbeats. It can be measured in the time domain, measures such as standard deviation or the root mean square successive difference, or in the frequency domain using spectral analysis. The vagal influences over the whole range of the cardiac spectra. Sympathetic influences roll off at about 0.15 hertz. So the higher variability in the yellow here is due almost exclusively to vagal influences, and that's primarily what I'm going to talk about today. This figure from 2005 just shows that the high frequency heart rate variability that we can measure non-invasively in humans, this is a rodent study, is related to vagus nerve activity measured directly. And Vaughan Macefield in Australia is now able to do these types of direct vagus nerve recordings in humans. So I'm trying to get him to come to California and show us how to actually do those measurements. So heart rate variability, we have published many papers over the years talking about heart rate variability and organism health, physiological health, emotional health, cognitive health. They're all interrelated. Again, these systems are not siloed off, but are interactive. The network of neural structures involved, this is a figure from Cor Wager and Richard Lane, showing the medial prefrontal brainstem axis. And some years ago, we published a meta-analysis showing the relationship between heart rate variability and a number of brain structures, in particular in the prefrontal cortex, the pregenial anterior cingulate, the subgenial anterior cingulate, primarily on the right side, the amygdala, primarily on the left side. So this is the basis for just an elaboration of what Darwin and Claude Bernard had spoken about. So in the context of well-being, imagine our ancestors walking along a path. They see something coiled in front of them, could be a harmless vine or a deadly snake. So what is the adaptive response? Well, it's to assume it's a snake. So Steve Julius proposed that this defense reaction may be a common denominator for hypertension. And I'll just read a couple of quotes from him just to give you a sense. So the sympathetic overactivity in hypertension reflects the chronic activation of the defense vigilance reaction. And the defense reaction may have been useful in evolution and may have offered survival advantage. In modern times, with prolonged life expectancy, the previously useful response in evolutionary terms contributes to a faster and deleterious wear and tear of the cardiovascular system. If these anticipatory reactions have offered a survival advantage in the course of evolution, they may have become a selection factor. The proportion of individuals with an efficient and prompt defense reaction could have selectively survived to pass on their genes to the next generation. In evolutionary terms, these mechanisms are geared to preserve the organism until the times of sexual maturity. What could have provided a relatively short-term advantage for the passage of genes across generations may have, under contemporary conditions of prolonged life, become deleterious to the organism. Now, this last sentence is the most important from my perspective. If we wish to further the life under modern circumstances, it may be necessary to dampen the defense response. So this defense response is what we call the default stress response, which is when in doubt, prepare for the worst. It's an adaptive response, it's fast and automatic, and is associated with the negativity bias in psychology, where threatening information is given processing priority by the brain. So just briefly to some data in support of this, this is a study published in 2001 with my friend, the late Jeff Ahern, a neurologist from University of Arizona. We did a pharmacological blockade of the prefrontal cortex in these patients undergoing preoperative evaluation for epilepsy, approximately half male, half female, half with the epilepsy on the left side and half with the epilepsy on the right side. Now, what's critical for our discussion is this prediction. So if the prefrontal cortex is tonically inhibiting these sympathetic excitatory threat circuits, if we take the prefrontal cortex offline, then we'll see an increase in heart rate, that is the default stress response, and a decrease in vaguely mediated heart rate variability. So these are the data. You see the baseline, and then over the course of the 10-minute inactivation, which was achieved by injecting sodium amobarbital into the left and right middle carotid arteries, you see that heart rate increases between 10 and 15 beats per minute. Now, if you're doing a study in your lab and you're doing a stress manipulation and the heart rate increased 10 to 15 beats per minute, you would say you had a successful stress manipulation. But what we did here was simply remove the prefrontal cortex inhibitory influence on these sympathetic excitatory circuits, suggesting this default stress response. Now, in the context of further elaborating on Claude Bernard and Darwin's work, my colleague Julian Koenig put together a large international study to look at the relationship between cortical thickness and heart rate variability. And this was in over 1,200 individuals. And what we found was that heart rate variability was positively associated with cortical thickness in a number of brain regions, in particular in the prefrontal cortex. And this is just a graphical presentation of that. So the larger your prefrontal cortex, the better you are able to have higher heart rate variability and essentially dampen the stress response. Now, this ties into this very interesting construct, this idea that Homo sapiens are self-domesticated creatures. And some of you may know that the Nobel Prize was recently awarded to an individual who's looking at the relationship between Neanderthals and Homo sapiens. So this is Homo sapiens, and this is very interesting. So this idea of self-domestication has some interesting implications. The idea that anatomically modern humans are domesticated species has long been entertained by preeminent scholars in biology and human sciences, in passing by Charles Darwin, and more seriously by Franz Boas. The domesticated traits exhibited by anatomically modern humans plausibly emerged following similar intraspecific selective pressures for pro-social behaviors. In other words, tameness towards fellow humans. Similarly, it's been claimed that reduced emotional reactivity and increased prosociality among humans are keys to our self-domestication. The self-domestication hypothesis has been a strong contender to account for key aspects of modern human cognition. This evidence that self-domestication can enable the development of complex behaviors beyond those discussed so far for the domestication syndrome. For example, both dogs and domesticated foxes outperform all non-human primates in tests of cooperative communication. The Bengalese finch, domesticated from its wild ancestor, the white-rumped munia, has developed a complex song that is preferred by both female finches and munias over the stereotyped song of the male munia. So this self-domestication may be a key factor. Now, how does this self-domestication manifest itself? In the context of wolves and dogs, you can see on the right-hand side the wolves, and on the left-hand side the dogs. And you can see that there's a change in the actual structure of the brain case, of the structure of the jaw, of the structure of the teeth. And when you look at Neanderthals compared to anatomically modern humans, you see similar changes. My friend Mats Fredriksson did a study looking at changes associated with domestication in rabbits and found, comparing wild rabbits and domestic rabbits, that using high resolution brain imaging, domestication reduced amygdala volume and enlarged medial prefrontal cortex volume, supporting that areas driving fear have lost volume, while areas modulating negative affect have gained volume during domestication. And then they close by saying that we propose that compared with their wild ancestors, domestic rabbits are less fearful and have an attenuated fight response because of these changes in brain architecture. This just simply shows those changes. So in the context of understanding this circuit of prefrontal cortex and amygdala, with my colleague Mara Mather, we published a study looking at amygdala prefrontal connectivity and its association with heart rate variability. And we found that in both older and younger individuals, the greater this connectivity of this prefrontal region with the amygdala, the greater their heart rate variability. In a study from some years ago, we looked at individuals that had damage to this medial prefrontal region. These individuals, as you can see, had damage to the exact regions that we're talking about. They performed a number of tasks, and we had 18 individuals with damage to this region in the prefrontal cortex, 12 individuals with damage to another brain region, and 54 non-brain damaged individuals. What we can see is that those individuals, the women that had damage to this prefrontal region, showed an exaggerated cortisol response to the tree or social stress task, and males that had damage to this region showed elevated heart rate across these tasks. Interestingly, for both males and females, individuals with damage to that region of the prefrontal cortex viewed the tree or social stress task as more threatening. So this brain region is important for recognizing safety and is important for modulating our physiological responses to stressors. So stress regulation is emotion regulation. One study from Rajita Sinha's group at Yale looked at this. So emotion regulation moderated the association between chronic stress and cardiovascular disease. So in this community-based study, they took a composite measure of chronic stress, a composite measure of cardiovascular disease risk, and they looked at whether an instrument called the difficulties in emotion regulation scale moderated this association. What they found was that for individuals with high emotion regulation, there was essentially no association between their stress scores and their composite cardiovascular disease risk. However, for those with low emotion regulation, greater stress was associated with greater cardiovascular disease risk, suggesting that the ability to effectively regulate one's emotions can dampen these defense vigilance reactions and modulate cardiovascular disease risk. This is just a verbal statement of what I just presented. With my colleague Dwayne Williams, we have shown that heart rate variability is related to the same emotion regulation scale. So we showed that individuals with higher heart rate variability have less difficulties in regulating their emotions, suggesting that heart rate variability may be a moderating and or mediating factor in this association between stress and disease risk. So we have looked at heart rate variability in the context of psychopathology, and I want to just finish up with a few studies here. I think I'll, Laura, how am I doing on time?
I think you're okay, although Emiliana is the formal timekeeper. I think you still have about 15 minutes. Is that right, Emiliana?
That's right. That's right. You're doing great, Julian. It's amazing.
Oh, excellent. Okay,
Yeah.
Good. Okay, so we have proposed that heart rate variability may be a transdiagnostic marker for psychopathology. A number of meta-analyses by our groups and others have shown that in general, both externalizing, internalizing, and in addition, thought disorders are typically associated with lower heart rate variability.